Okay, so we stopped here. So the first issue that we have, so we have three methods here. I mean, two methods, two generic prediction method, and one and Web 2, and we have a Q3, so a three-state prediction method. So one in what's the fraction of correctly predicted in either of those three states, and there's a random background. And so did you see that? Did we discuss that already? Yeah. So the first question is, when you look at two numbers, the first one is what is random. So it could be 33. It may not be 33 because we have three states. Why would it not be 33? Did we discuss that already? No? Okay, so it's a valid question. As long as I see question marks in your faces, then I'm on the right topic. So why would it not be 33? So we have a three-state model. I say random is 33, and then I ask you, why would it not be 33? What could be the reason for random to be different than 33 for three-state? Yeah?
Maybe if the classes aren't balanced?
Exactly, if they're very uneven. So, for instance, in the simplest case of secondary structure prediction, where half is sort of the other state, 30% is helix, and 20% is strand, in that particular case, if you randomly shuffle it, you get 35. Okay? Now, or let's put it differently. If you sort of tweaked your probability, what would you do in this model? So you want to have the highest Q3 score in the distribution that I just said. You have 50% other, 30% helix, 20% strand. You have to predict one of these three, and I give you sort of what do you do? Yeah, I believe you got the solution.
You just predict the helix class.
Yeah, just in fact in this particular case, so in that sense this is not quite random, but the bottom of the case here, you have to beat the dumbest prediction, or you want to predict the dumbest prediction, beat the dumbest prediction, which would be majority class only, which would be 50%, right? On the upper end there's also an issue. The prediction is not 100% because you make experimental errors. And you cannot learn more than so you even very good machine learning will not learn better than the noise in the data, or will hardly ever learn better than the noise in the data. So it's a reasonable statement to assume that you will not get better than the experimental noise. So if the experimental error is 5%, then 95% is your ceiling. There are other ways in which you sort of can work out the ceiling. The ceiling could be, so how would you measure the experimental error? Well, the experimental error, one way of doing it is two experiments agree to a certain degree, right? Or what else could you do as an experimental error? So one is essentially that two methods agree, or two experimental methods agree, NMR and X-ray crystallography. You could do 2 times the same X-ray crystallography. What else could you do to sort of gauge the experimental error in a biological system? So for instance, you want to measure subcell localization. Okay? In this particular case, it's not done by X-ray crystallography. It's typically done by a GFP tag. So you have a green fluorescent protein that emits light when it's reported. You see that, or you don't. So one way to gauge experimental error is you look at the agreement between two different experiments. What else could you do in a biological system? We're talking about proteins. Any idea? Well, we can, as a statistical set, look at homologs. So in the context of this course, we have always talked about homology-based inference as a way to predict. But you, in some sense, can also see that as a way to look at the reliability of a system. Well, that may not work between mouse and man, but it clearly, or most likely, will work between two people. So most proteins that between you and your neighbor vary will still be localized in the same place, right? In some sense, that is also a way to gauge the experimental accuracy. And that, in this particular case, you would gauge the experimental accuracy in a way that is a little bit independent of sequence, because you vary sequence. That's another way of looking at it. Okay, anyway, so you have a top, you have a lower, and you have your methods in between these two. And now comes the question, which method is best? Say in this particular case here, which method is best? How do you rank them? What do you need? By the score. Absolutely true, Benjamin. There's no other. I mean, you're not going to sort of invent another random score and rank them by the score. So you're going to sort your things by the score, right? The absolute trivial thing. Numerically, this is the highest. This is the second highest, and this is the third highest. But now comes the point: is that implying that this actually that you can show with this table that this number is the right, that this in fact is the best method? No, you cannot. What is missing?
Have the scores been normalized already?
I'm not entirely sure what you mean by normalized. So we are comparing.
The same data set. No, that was what we had.
That's an important issue, very, very important issue that Benjamin brings up. Before we can, you will find in publications many tables of this type, where sometimes there's an additional star that says data set picked from that publication, here 2 stars, data set picked from that publication. That already is a no-no. In many tables you will not see these numbers coming from the same data set. They have to, otherwise you cannot compare them on the same table. Do not put things on the same table unless they really compare apples and apples. In this particular case, that is already the case, and that's what Benjamin meant by normalized. So the data set is already the same. But again, we took care of. But there's something else that we are still not showing here that you need in order to make that decision. So let's assume in this particular case the standard error were 0.8. Okay? So standard error being 0.8. And I have sort of 5 methods in this particular case, where again, in numerical sorting, B is the highest number. My question to you is, if I now rank it, so I believe, no, that already is a solution. So if I publish a rank, and that rank is one where I say I will put into rank 1 only the method that is statistically significantly better than the other one. Okay. Clearly, this will be in the first rank. What else will be in the first rank? Anything else?
Yeah, A would be first rank too.
Okay, so we will have A and B. That's it? So numerically, the next number is this one here. 0.8. Let's just assume one standard error is enough. So let's look at C. The 0.8, okay, now if we really talk about one standard error, this is more than 0.8 away. Don't take my attack or my trying to trip you for, trust your own. The standard error overlap, that is the first important issue. So what I'm doing as a con man here is I'm sort of injecting in you something that we sort of took as accepted territory, but the standard errors overlap. So this already is not a clear decision. But there's something else that makes it complicated. So the standard error between C and B overlap in the sense that if I add a standard error here and I subtract a standard error, we come to points that overlap. That's what Natalie says, and that's right. It's still technically like almost 2 standard errors. So, but this is an issue. Natalie's totally right. But there's something more relevant as an issue here. Do not look at B. And also don't look at E. at this point at least. Anybody catches the problem that is? Okay, I'm saying that B ranks 1, okay? Because I cannot distinguish between A and B. Not with the 2, but between A and B, this is clearly within 1 difference, right? That is a very clear too close. Okay, if I cannot distinguish A from B, then the next one in rank 1, being C, well, look at these 2. Right? So A is somehow the middleman in some sense. And the middleman that pulls in C. And if C is pulled in, you immediately see that D is pulled in. So that creates the strange situation where in rank 1 I have 4 methods. Although I can clearly distinguish D and B, the decision that A slips in with B, and there's no way around it with the standard error, makes sort of C slip into that. And then essentially I create ABCD as a group that I cannot distinguish, and I create the only thing that I really can distinguish from all of those is E. Yes? Benjamin?
Yeah, but there is a clear distinction between C and D.
That's the point. That's the point.
Because C is, while it is 1 standard error away from so while it overlaps 1 standard error from B and A, that only factors in if it's the plus standard error, right?
But the point is, if you make the point that B is rank 1, C, I should have inverted it here, that A, although it's the second best here, is in rank 1, that C and A are too close. So you all feel this, you all accept, and I'm not being con man, you all accept that in the strongest way of pushing this through, where you in fact always become part of that group, provided the next is not distinguishable from the last you put in. And there is something you can argue about it. Can you think about any other way in which you sort of could construct a rank? Again, there's the numerical sorting. But the numerical sorting has this issue, it is not considering statistical significance. So the numerical sorting is not the way out. Okay? This may not be the right thing, but numerical sorting is always worse. If you just say that this numerically has the highest number, and if you cannot distinguish from the next highest number, then this highest number is not meaningful. And you should actually not rank it. But here's the alternative in which you could do that. You could just sort the matrix here, and you could ask for every single method, let's begin with A, that the diagonal falls away. So I'm going to put a green one in here if A is significantly better than E. If A is significantly better than D. If A is not significantly better than B, or significantly worse, then it will be red. But that I cannot say. So I have two white ones here, and so forth and so on. And here I have some red ones, read it in that direction. D is significantly worse than A. D is significantly worse than B. Do you now get a way of ranking? How can you translate that to a ranking? It's obvious, no? Yeah.
Yeah, just look at how often the method is the best one.
Yeah. That's one way of doing it. And it's a very clean way of doing it, because you now clearly can show that B in fact is better than 3 other methods, while A is only better than 2 methods. Although I cannot clearly say that B is better than A given the standard error. Overall, for the entire set that I have, I get a ranking scheme that is consistent with the idea that B actually is the top performer, because it beats other methods statistically significantly more often than A. Okay? Again, I can still not say statistically significant A on a one-to-one comparison. A and B are continuing to be blank, not statistically significant. But still, B outperforms A more often, or outperforms the other methods in my comparison more often. So in this class comparison, the ranking now becomes clear, and this ranking becomes one that is essentially very, very, very similar. And it happens to be the way that it is constructed, very similar to the sorting that Benjamin sort of suggested in the first place. But this is not what you typically get, or very often. Just be very careful with this numeric sorting. So this is one way out. Everybody clear about this? Or
But shouldn't it be symmetrical?
say again?
Shouldn't it be symmetrical?
Did I make a mistake? Absolutely, it should absolutely be symmetrical. And there is a red one here that is missing, is what you're saying, right? Thank you very much for being awake. So that means I can clearly close the window, because you're feeling cold and you're completely there. So yes, so this should be red. Totally symmetric. Other than that, everybody clear? Yeah.
Yeah, but if we use this matrix, basically all we do is we use the numerical values to ascertain which method outperforms which other, and we do not take into account the standard error. So
yes, we do. Because, this is why it's white. The white ones are in fact the ones, so A numerically is worse than B. But that is not statistically significant.
Mm-hmm.
So that's why there are three colors here. I mean, again, ignoring the diagonal, there are three colors. There is...
I see. Yeah.
Okay?
Yeah.
That's the very crucial part. Yes, we do.
Because there's an overlap to the minus antenna, like there's a two-way overlap with A and B, but there's only a one-way overlap with B and C.
Yeah.
Okay.
Exactly. Which again, that really depends on the numbers, how these numbers play out in the end. In this particular case, it plays out that way, yes. Okay, so all of this is sort of simple as long as we have a distribution such as this. So we have some performance, the Q3 or whatever the number is, that I happen to call in the first one, and then there's a distribution, so a number of, in this particular case here, proteins that have a certain Q3, and you see sort of an average, and that was the average that the whole thing was played on, and then there is a standard deviation that leads to a standard error. Everything is clear. Now, if I predicted subcellular localization, I get a distribution like this. For some proteins here, blue, it will be right, and for some other proteins it will be wrong. I can have a subcellular localization predicted correctly, yes or no. There is no subcellular localization predicted correctly for 20% of my protein. The protein is nuclear or is not nuclear. It's a totally binary thing, right? By the way, back to sort of quickly parsing in your brain numbers and things you see. So I'm claiming that on the right hand what you see is 60% accuracy, two-state performance. How many of you immediately jump up and say that it's not true? Nobody? Only one? Are you jumping up, or are you just smiling because I asked?
It's like 40 or something.
It's certainly not 60, right? So when you see a statement like that, immediately, 60% means blue is higher than red. Well, before you ever look at the numbers, blue is not higher than red. Okay, this is the first thing. And then you jump a step ahead and you actually looked at the actual numbers. But that's the detail. The first thing, when you hear 60, 50, whatever these numbers, look at, are these the same, right? Look. Anyway, back to that. Now the question is, how do I get from such a binary distribution, how can I get a standard error? That's the question. Any idea? No one? So binary, yeah?
Yeah, you could split the data set into many small ones and test on each of them.
So we take our data set. So going back to slides. Oh, I have it here. So say the numbers here is 2,000, 1,500. Felix says about 40%. I'm not sure. I'm too lazy to actually count it up now. Say this is sort of roughly 40%. That 40% you get from your data set D, and just take everything in that data set. So for all the proteins for which you predict, you know whether you're blue, right, correctly predicted, or red, incorrectly predicted, right? So you have you have a table for, say this is 1,000 proteins here. Then you have a table of pairs, the protein identifier, and then you have true or not true. So something like that, right? You have 1 or 0. In the image above here, you have a blue or red, okay? Now what you're going to do is you're going to take out a protein, put it onto a stack here, and put it back. Take out another one, put it back. And you do that 1,000 times. So you create a new data set that has the same size as the previous one. It's not identical, because you randomly choose. So it could be, so if you randomly do that, very, very often you will have the same one more than once in here. Right? Just randomly. So you very, very, very probable that if you pick 1,000 times at random, you will not take the same 1,000. It's extremely unlikely that you will just exactly get 1,000. That you will get really all the 1,000. You will most likely pick the same one more than once. Okay? And then you will create a data set. And then you do that again. And again. And again. And you do that, say, 1,000 times. So you do 1,000,000 picks. That doesn't take any CPU, because ultimately you just pick at random from your table that has the labels already in there. Right? And you just recompute the average. And that average that you get, that ultimately is the standard error. Okay? The technique is referred to as bootstrapping. It is initially published by Bradley Efron in the 1970s, 80s, early 80s. And the image that comes to my mind when I think about this bootstrapping is this Münchhausen story here. Some of you may know the Münchhausen story, who is pulling himself out of the swamp. So he sits on a horse, he rides on a horse, and the horse and they fall into a swamp, and then he pulls himself out of this swamp, including the horse. So that is the Baron Münchhausen is the guy who sort of tells stories that are a little bit fantastic. But anyway, in some sense, this is exactly what bootstrapping does, is you use the same data. You just have reproductions of the same data, and you get an error or a distribution out of something that doesn't have a distribution, because it's a binary distribution, right? And again, sort of the standard deviation in this particular case becomes the standard error. Now, this was statistical significance. What else is there? So statistical significance, again, is you have a distribution, and you want essentially to say that two distributions, they differ, yes or no. That's statistical significance. When they don't overlap, they're statistically significantly different. What else could we look at when we ask whether one method is better than the other? So we get a different number, and say the different number is such that I say method A is significantly better than method B. What else could I look at? Yeah?
Or with one specific state you're interested in.
That is true. So ultimately what Daniel says, and that's a very important one, so far I'm talking about Q3, maybe that a particular method that has, so where A is better than, AQ3 is better than BQ3, but maybe A is worse in predicting helices. And what I care for in my nuclear proteins, what I care for would be nuclear proteins or something like that. That's really important. But before I get into that point, there's something else. Something more more substantial, on a higher level of perspective. So again, I can say they are statistically significantly different. What else do I have to establish, or do I want to establish, or? It goes in a similar direction, but I mean something slightly different. So saying it would be something completely different from what you say, Daniel, would be misleading you. But it's not quite that. Okay, let me ask it differently. So if I replace the word statistical, by what would I replace it? So I keep the word significance.
Biological.
Exactly. So I would sort of replace it by the word, but essentially that is what you say. I would use an S because, in a more general way, scientific. But in this particular case, it's biology, of course. So Daniel's totally right. Biologically significant. And that, what does it mean? So somebody else. So he guessed the word. Now guess what it means. Somebody else. Any idea? Ultimately, it means that the difference that I have in performance here, say I have an 18 class difference of 5 percentage points. What does it help me in terms of biology? What does it allow me to do that I couldn't do with method B? Is there anything, any kind of experiment I can now get an answer for that I couldn't do with the other one? Is there any kind of using the data, for instance, looking at all the nuclear proteins in human? Does it help me, this improved performance, to do something that I couldn't do before? That would be scientific significance. And that is very difficult to establish. So one way in which you can establish it is you have a collaborator, and the collaborator tells you, for the previous method, I never discovered my protein because the error rate was too high. For the method that you have now, I managed to discover that. This would be an example where you could sort of have a proof of concept of scientific significance. You discovered something, or your improvement led to a discovery. But it's a very rare case. Anyway, so I want to make you aware that there is a different issue here, and if you can, you clearly have to find a way to address that in your publication. It may be speculation. It may be such that you say, in the end, I established that the method is statistically significantly better, but this is not, I cannot establish that it's scientifically different. Could be, but you have to still address it somehow, okay? Let's get back to the story of subcellular localization, where we have this 18 class prediction, and again on the highest level here we distinguish between proteins that are soluble and transmembrane, and that is done at a level of 94% accuracy, which sort of is similar to the best state-of-the-art method, Polyphobius, that is a specialist on that. And in fact, I'm not entirely sure why we didn't take Polyphobius if that is higher. I believe at some point we convinced ourselves that we are even higher than this. Anyway, so in principle, by the way, if you found something like this, then essentially what you should do is redo your method and use this method, this tool here. Although it is ±2, so you cannot really establish that these two methods differ, but your first impulse may be the right one on a situation like that. Take the way Polyphobius to make that first decision. And then we go on further down. When we go further down, these numbers decrease. Why? So you can also tell me that when you look at detail, you see some of them are still fairly high. In fact, they don't decrease. Here's 80% and there's 44. So some of these numbers do not decrease. But that is true. There are exceptions. But overall, there's a tendency that these numbers, the more you go down a particular path of the tree, the more the number decreases. And yeah?
Yeah, the accuracies add up from the top to the bottom.
So not strictly in terms of they don't add up to 1.
Yeah, but if you make a decision and this isn't 100% correct, you add up this error in every step?
That is the first most important statement, yes. So the error adds up. Any mistake you make up here, you cannot recover from. That's one reality. Another reality is, the further you go down here into detailed decisions, the more challenging the problem becomes. The design of this hierarchy was, remember I said, you try, since you cannot recover from errors, you try to have the best decision up there. That means the things where you do worse, you try to have somewhere down there. Which means you try to put the problems that are the most difficult one somewhere down there, and that's why you find the most difficult ones are down there. Okay, so then one of those issues that I always tell you is, when you do a thesis, you have to sort of take all your data, reduce it to as little as you believe is really important to make your point that you're doing better than something else. And you have to always compare your method to somebody else, okay? Now, that typically means that you have as few scores as possible. Daniel already said that we have to look at more than one score. Just looking at the Q3 may not be enough. On the next slide, I'm going to look at just one score and already have a complicated slide. In this complicated slide, this is bacteria, but let's forget about those at the moment. Here's only eukaryote. Eukaryote only. I told you our system predicts 18 different classes. In fact, I showed you on the slide before here, those are 18 different classes. This one says Q9, which means 9 classes. So why do I compare two methods here based on 9 classes? You cannot know, yeah?
Yeah, maybe the other method only predicts 9 classes.
Exactly.
Only compares.
And this is why, in fact, this one here has Q5, because that, in fact, is the first version of the method that we had before. It uses the difference between these two here, that is based on neural networks, that's based on SVM. That uses overall protein features. This one here uses the profile kernel. So we're looking at the k-mers, the conserved k-mers. That's the idea we discussed last week. And so we have an improvement from 62 to 66. When you look at the error, you see this is not significant. Okay, the same is true here. And in fact, for the moment, let us look here at the one of these, either of those, but we can look at this one here. This is bacteria, and you have a difference between 72 and 86 with a standard error of 18, and therefore I cannot say that this method is worse. Right? Yes?
Should that number, like the score, should it ultimately add up to at most 100?
Yes. Oh yeah, yeah. So there is an issue. There's always an issue with this plus minus. But so an alternative way to do this. Can anybody suggest to me how? So Benjamin puts his finger on something that is absolutely right here, right?
Also there and.
Well, but let's just stay here. Wherever you put. So the first mistake is here. Or the first mistake is there, wherever you look first. The other two actually work, but here it's wrong. So 86 plus 18 is a number that you don't want to write. How can you avoid that? If you want to have a table in which you don't suggest that you believe numbers above 100% make sense. Which in fact, again, what Benjamin says here, you have to do that to your own work. Just hold on. There's some problem, and in a table in which you show that, you clearly should do this. Hold on. It does not mean that I believe there's over 100 possible. I'm just doing that because plus minus is a symmetry. What else could you do? Well, you could sort of write the range, right? So the number is 86 plus minus 18. So you say the performance is between 100 and 86 minus 70. Oh, I'm slow. 78, 68. So you put that into the table, right? Then you get rid of this statement or this mistake that you actually claim something to be above 100%. You say that my estimated performance is between 68 and 100. The problem with that is that you're actually moving the way you construct the interval, you then would have to somehow say, and my average is 86, and then it becomes unwieldy. Because it's no longer symmetric, right? So for normally, if you do that sort of thing, here, you would immediately compute where the average is. If you just publish the numbers like that, people would sort of intuitively say, okay, the difference is 32. 32 by 2 is 16, so 16 minus 100, so 84. Which is not the case. That's the downside to this. Anyway, if you look at the box plot, yeah?
But if it's the standard error, right?
Yeah.
The standard error is symmetrical.
That's why it's plus minus.
Yes, sure, but then it should be 84 and 16.
No, but so we are clear about the average. 86, not 84. And since the standard error should be symmetric, it's written as plus minus 18. What you can also write plus 14 and minus 18. But then whatever you do in a case like that, people will trip.
All right.
So then they will say, well, how come you define non-symmetric standard error? But then again, as long as you sort of point out in a caption to the table what the reason is. And in this particular case, you should actually point that out. So I believe we committed the mistake of not pointing it out in this particular case, and people stumbled over it. Anyway, so partially I showed this to say that even simple things, one single number, one single score, can get difficult if you do the right thing. So be careful how you quote them. That needs to be in a table in your publication. That clearly needs to be somewhere up front in your thesis. But this is not the kind of thing that is easy to convene to people. You're doing better. And too many numbers are clearly confusing, and I believe this is already at the point where we have too many numbers. So this is already at the point where you for yourself can look at this and you can make statements, and you will have supporting online materials, appendix for a publication, but this most likely already will not get, may not get to the mainstream, although clearly you have to present a comparison of your method to something else. So it is a tricky business. Okay, there was this other issue that I said, the kernel, the idea of the profile kernel was also that we want to be not too sensitive to sequencing mistakes. So one of the problems of the Loc Tree that I showed you before is it takes the amino acid sequence, the amino acid composition of the entire protein. If I make a sequencing mistake, then I have something else, right? And here we simulate something else in three different ways. One is we remove the first 30 residues, the N-terminal. We remove the last 30, and we remove randomly 1/3 of the protein. Which, mind you, since most proteins are longer than 3 times 30, 90 residues, this removal here is going to remove more residues than the other two. Okay? And randomly means just one region. So I'm not randomly picking some residues that I remove. I randomly pick a region of 1/3 and I remove. Okay? And then we see comparisons here. The standard error is much lower now because essentially we can only compare numbers in that direction. So the methods are not intercomparable. They don't use the same data sets. So the standard errors were so high on the previous slide because we needed to find a new data set that none of the methods had ever used before. Here we don't need that. So they're comparable within each row. Now the first thing you see for all of these three methods here is if you take the full length sequence, you do the best performance. So they all get worse by cutting out things. The next thing you observe, however, is that there's a differential behavior. So while these two methods here do worst for removing the N-terminal, the first method here does worst for the last. A little bit. So we cannot, again, plus minus 2, we cannot quite distinguish that, this number here from the last one, but that's at least a tendency. Let's begin with this. So why is it so detrimental to remove the first 30 and not the last 30? Yeah?
Because most of the localization signals are located there.
Many localization signals that we know of are located there. It is not quite most. So what we know is localized there is the secretion, the one for secretion, for chloroplast and for mitochondria, which in fact together do not make up most of the proteins. In fact, I just want to repeat, most localization signals we don't know. Okay? So most localization signals, if they actually exist, are not covered by these two. But at least those, clearly the ones we know, and clearly they make up an important signal, and that is most likely what we see here. Okay. Now, why do you believe for the profile kernel-based K-mer method would this be such a big hit? And why is it not so much a big hit for the others? How would you interpret? So this interpretation, this is not. So far what I did was result. What I'm asking you now is to do the discussion. So speculation. So your guess is as good as mine, maybe. Maybe mine is a little bit better because I've done it a couple of times. Again, so what we learn is that these two methods here heavily rely on something that is in the internal signal. So they clearly use signal peptide prediction. And that is why there's such a rapid drop by removing it. If I randomly remove 1/3 of the protein many times, I will keep the signal peptide. This is most likely why that number for those two methods is still fairly high. For Loc Tree2, that is not the case because Loc Tree2 essentially looks at K-mer and they're distributed throughout the sequence. This one here removes much more than the others. Yes, so Loc Tree2 somehow learns that the internal signal is sort of more important. But then ultimately, this is balanced by the fact that removing more of these K-mers is also not good. So again, this removes physically much more part of the protein than this. This much more than these two. Only 30 residues in each. If an average protein is 400 residues long, 1/3 of that is 100 residues. So 3 times more, roughly, right? Or 3 plus, 3, whatever. 4 times more. And that ultimately is what we most likely see here. And what we also see here on the entire table, what you sort of can appreciate, is that relatively, if you look at what tool is stable against the removal, then this one is relatively stable. So to some extent, yes, so moving part of the sequence does hurt, but the prediction is still possible, even when removing sort of 1/3. Okay, so the next issue is, I've said we want a de novo prediction that beats that beats homology-based inference. Now, typically, what you do is you put up your server and you say this is the de novo method, and user, if you know you can apply homology-based inference, do it. But that turns out to be not a good idea. It's better if you could put homology-based inference into your system. How would you do that? So we have the de novo method now. We call it Loc Tree2. And we want to develop a new method that simply combines homology-based inference with a de novo. We'll call that Loc Tree3. How would you do that? What's the simplest way of doing it?
Markov chains.
That sure is not the simplest way of doing it. Is it?
It was the first one I could think of.
Well, I thought the first one you can program. And if you had to sit down and program it, is that the way Markov chain, you have to sort of go back into, so you mean hidden Markov models, right?
Yes.
That's not that easy to train. So you may have a library, but then you have to do the retraining. Something simpler. And in fact, it may be the right thing to do. We haven't done it. And again, the reason why we didn't do it is because very often in these cases, somebody says your deadline is on Friday this week, and you still have to do things before you actually write up your manuscript or your thesis or whatever it is. And suddenly somebody comes and says, on top, you have to put that in there. So you're not going to do a hidden Markov model. You're just counting how many hours sleep am I actually going to get. None, but no food, but I still can't manage. And then clearly the answer will not be hidden Markov model. What could you do? Yeah?
You could have profile information.
How?
Yeah, by multiple sequence alignments and then just looking how often that amino acid is occurring at that position.
But what you do then is you do a prediction method. You develop a new prediction method, no? Loc Tree2, whatever you call that.
Yes.
No, you don't want to get into redo your thesis. So you imagine that you worked on your thesis for, in this particular case, it was more than 6 months. And now you're given a few days. Literally, it was 1 day in this particular case.
I would do a normal prediction and then search for homologs, and if there are experimental annotations that overlap with my prediction, I have additional support, and otherwise I could maybe use.
What do you mean by additional support? You have 2 annotations and you report them both. That's a good idea. Is that what you meant?
I think not.
No, it's not what you meant.
So if the annotation of the homolog is the same as what my prediction method predicts,
You're happy and you return that. And if it's not?
happy, and if the homolog has another annotation, I might use it anyway.
Okay, so this is what I thought you would say. So typically homology-based inference means that if my sequence similarity between a query and an experimentally annotated protein is higher than some threshold t, I say that I can look up the annotation from E to Q, right? That's homology-based inference. So that means in this particular case, I have the threshold, and we simply say that if I find a homolog just the way Latterly says. If I find a homolog, I take it, and if not, I run my de novo prediction. Okay. Here is where that gets you. And let's play this and continue with everybody present. So again, here is one way of showing the final result. In this particular case here, in the image you see that green is predicted, so it's predicted to be nuclear. And for every single decision here, it's difficult to read that. There's a probability of making that decision. And there's a probability with a final decision, and the final decision is ultimately the probability that is used here. Now, what you can do, since you can predict subset localization for an entire organism, or for every single protein, you can do it for an entire organism. And this is the first prediction that we got. Can somebody stumble over something?
Yeah, it's weird that the distribution of classes is so different between human and orangutan. You would think that it...
Even worse, if you sort of look at, just look at, for simplicity, let's look at nuclear. Human looks much more like a chicken for the nuclear protein than like an orangutan. That's odd. And there's a variety of reasons why that is the case. Now, the first one, I want to get it with a slightly different issue, because I believe it's simpler to make my point on this issue. Let's assume the task would be to predict how many membrane proteins are in human. Human has 20,000 proteins, and say my prediction is this. So 5,000 are predicted by my method, 5,000 of these 20,000 are predicted to be transmembrane, and 15,000 are predicted to be non-transmembrane. Now I ask you, what's the fraction of membrane proteins?
25%.
Is that what you were saying?
30.
Boo. So Felix just go to sleep for the next 5 minutes until I get to that number. Anybody else?
It is 30.
It is 30%, yes. And everybody who understands why it's 30, you can also go to sleep. Everybody not. So the question is that really the best you can do? But you could actually not have known that, so you're cheating.
I know that 30% of the proteins are in membrane.
No, actually, that's also not true. I don't believe it's true. But oh, is that why you? Then you have to wake up again. Yeah, it's true. It is sort of you answer the question in a different way. Let's just assume I have a hypothetical problem, and just consider to call two types of proteins, T and N. They have nothing to do with transmembrane. It's just a total mistake from me that I put the word transmembrane up there. What is the mistake in concluding 25 here? What could you do that is better? And Felix didn't do it, so Felix cannot help you.
N is for nuclear?
He used a trick.
N is for nuclear?
No, N is for not T. I could have called it R or whatever. I'm sorry. I'm sorry to be misleading. In my last version, I always had a not T, and then people stumbled over what this not symbol. So now I put it into an N. Whatever I do is wrong. So just I'm not sure how familiar. Whatever you believe, how familiar you are. The last time I gave this lecture, people stumbled over that one. Okay, so what is missing here? What could you do better? You have to think about the error. That is your error. Whenever I want to hear the word error, you don't have the word error. But in this particular case, it's not standard error. I'm not after standard error. I'm after something else. I'm after the mistake you make in your prediction. Say, I have a 10% error for both of my classes, okay? So, of the 5,000 membrane proteins that I predict, or T proteins that I predict, 500 are not T because I have 10% mistake, okay? Of the 15,000 non-N, I also make 10% mistake, so it's 1,500 that are a mistake. So, that actually means that I predict 6,000 T's. Is the 5,000 minus 500 plus 1,500. 6,000. And I predict 14,000 N's. And that, of course, is the 30% that you talked about. That's exactly your 30%. Again, so there's I do not know what so the people who came up with a 30% number used the wrong genome, they used the wrong prediction method, and they did not think about this issue. So there are many mistakes how the 30 comes about. I do not remember what the current number is. At some point, there is a publication that I believe quotes it, but I don't believe it's 30%. Maybe you're right, but I seem to. Anyway, this is something that you have to always do. If you write that in terms of formulas, it looks like this, where this, in fact, here is the confusion matrix. The confusion matrix, you're all aware what the confusion matrix is? Yeah. It's just what I showed on this for the T and N. It's the same story. There's nothing intelligent in here. It's a totally trivial. That's Valérie Marot. Yes, you recognize her. Oh, yeah. So in fact, that is Valérie's bachelor thesis. That in fact is not true. Is published by now in bioinformatics. So. The math is really straightforward. There's nothing, as long as you understand the story that I showed you with the T and N, you can work out for whatever time it takes you, in a few minutes, in a half an hour or something like that, you can work out the math. It just looks way more complicated than it actually is. It's really simple. Okay, and then you can now look at, for instance, 3 different prediction methods and ask how well do they reproduce some experimental data set that you have. This one happens to be called Protein Atlas, whatever it's called. It's not important. You see that white would be there's no difference between the prediction method and the final data set. Red means there's a big difference. So the first thing you see here is that 3 different methods differ in terms of how good they reproduce the reality. On the second one, I have the corrected version. So it's the same data, it's the same prediction method, just corrected by the mistake they make. And now first of all, you see that they are very similar to each other. Secondly, you see that they all get whiter. So the mistake they all make in getting the final answer is reduced substantially. And then the other thing that you see that actually two classes sort of become very, very similar. But that's a different story. Now I can use that to look at the entire organism and predict again the subcellular localization. Now I don't have it on a pie, I just have it in this direction here, where these numbers give the percentage of a particular class. Unfortunately, the downside to this presentation is I no longer know what these numbers are. Most likely this is secreted. Most likely this red one here is nuclear. Now you can use these percentages here and compile a distance vector and use the distance vector to make a tree. On this tree here, you see sort of some of the features that you would expect. And now it's no longer orangutan, it's the chimp and gorilla, human. And you see that they are now much closer in the tree than they are to, oh, the chicken is gone. Okay, I cannot make the chicken point anymore. But even the plant is also gone. What was on the other one? There was fish. It's also not on here. Yeast. Well, yeast was different. Okay, I cannot quite make the point to the previous slide. But you do see that they are close to each other, that these two are close to each other, that they are sort of falling into a group that is separated from these. What you see here is that mosquito and fly fall together. And again, this classification here makes sense biologically, and all you ever do is you look at the overall composition of proteins predicted to be in either of these 18 subcellular localizations, right? There's some information that is lost because you present the data here on 1D. If you presented it on 2D, PCA, principal component analysis, you would see a little bit more. Now you see that still, I mean, clearly the mammals here are grouped. But you also see that there in fact is, on this axis, on the y-axis here, there's a large difference between these two yeast species. The two yeast species are very different from everything else, but they also differ from each other. And you see something similar in some sense here for rat and mouse. So rat and mouse are sort of most similar, so the tree version is sort of reflective of that. But you see that there is a substantial difference, and by some ways clearly chimp and gorilla and human are closer to each other than these two. And then you see essentially what you get over the tree here is you get an additional resolution.